Hello, my name is Kai Kober, and I am presenting prediction of CDK4/6 inhibitor outcomes in metastatic breast cancer patients using exosomal microRNAs. Some background. Breast cancer accounts for 1 in 3 of all new female cancers annually in the US. One common treatment for metastatic breast cancers are CDK4/6 inhibitors. However, some patients develop resistance to this treatment, and it is not understood why that is. Exosomal microRNAs, being mediators of communication between cells, may improve our understanding of the mechanisms underlying treatment response and resistance. This being the case, our study focuses on determining microRNAs and their biological pathways as predictors of treatment response for CDK4/6 inhibitors in metastatic breast cancer patients. We assessed 11 patients who received CDK4/6 inhibitor treatment, 5 of whom responded to treatment and 6 of whom did not. Each had their microRNAs analyzed for differential expression comparing the treatment-responsive and treatment-resistant metastatic breast cancer patients. This resulted in 54 microRNAs associated with treatment response and 18 microRNAs associated with treatment resistance, both of which had p-values of less than 0.001 after adjusting for race. We performed a network analysis of microRNA gene target relationships to visualize the interconnectivity of the previously assessed microRNAs to their targets. Then we performed an enrichment analysis of target genes to determine which pathways were statistically significant. With these pathways in mind, we found a review paper that described several hypothesized resistance mechanisms of CDK4/6 inhibitors. Comparing the pathways found in the enrichment to those in the paper, we identified several enriched pathways supporting a number of the proposed mechanisms of resistance. There weren't any pathways that were exclusive to treatment resistance, but 3 mechanisms were exclusive to treatment response. This slide shows the comparisons of network instructions for treatment resistance and treatment response, showing the interconnectivity of microRNAs in the blue boxes to their respective gene targets, the purple dots. The treatment response network on the left has many more connections to that of resistance, possibly explaining the 3 mechanisms exclusive to treatment response. In conclusion, pathway analyses of the differentially expressed microRNAs support several hypothesized mechanisms of CDK4/6 inhibitor resistance. This being the case, the study demonstrated that it is feasible to utilize exosomal microRNAs to predict treatment outcomes in metastatic breast cancer patients using CDK4/6 inhibitors. Future studies could utilize a larger patient sample size, and therefore potentially more differentially expressed microRNAs, to determine pathways exclusive to treatment response or resistance for more conclusive evidence. Thank you to Dr. Chan and everyone else who contributed to this presentation, and thank you for listening.